Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine biting into an apple and, not having to worry about that twinge from an old filling.
Yeah.
Or even better, maybe never needing a filling at all because your tooth could just repair itself.
Yeah, sounds like science fiction, doesn't it?
Totally. But scientists are actually making some pretty big strides that could make this real. And today we're doing a deep dive into some really fascinating new research.
That's right. We're looking at a study from King's College London and Imperial College London. Their work, it was just published in ACS Macro Letters, and it details how they've been, creating an environment in the lab.
Environment.
Yeah, one that can actually support the growth of tooth structures. So we've got a news article summary, but also the original scientific paper itself to really, dig into the details.
Perfect. So our goal here is to really understand this potential breakthrough, right? What it might mean for the future of dental care, but also let's be real about the challenges, the hurdles still ahead.
Depth.
So let's start right there. Why focus on growing teeth in a lab anyway? I mean, what's wrong with how we deal with fillings and stuff now?
Well, that's a really good place to start. Current treatments, things like fillings, implants, they're common, sure, but they aren't perfect, especially long-term.
Right.
Fillings, for instance, the paper points this out, they can actually weaken the tooth structure over time. They don't last forever.
No kidding.
And sometimes, unfortunately, you can get more decay right around the edges of the filling itself.
Oh, yeah. It's like a patch job, isn't it? Fix one thing, maybe cause another issue later.
Yeah.
You're back in the chair.
Exactly. And then you've got dental implants. Now, they can be a much more permanent fix for a missing tooth, but it involves surgery. And crucially, they don't really replicate the natural function and integration of a real tooth. They're fundamentally artificial.
Okay, so the big dream then is what they call biological tooth replacement, actually regenerating a whole new tooth.
That's the ultimate goal, yes. The vision is to regenerate a complete tooth that would, naturally mesh with the bone. To do that, scientists basically have to copy how teeth develop naturally.
Okay.
And that means replicating the interactions between two really key cell types, the dental epithelium and the mesenchyme. Those are the main players in tooth formation.
Dental epithelium and mesenchyme.
Yeah.
Right. So those are the cells doing the work when a tooth normally grows.
Exactly. And previous attempts to grow teeth in a lab, they often ran into trouble trying to deliver the signals that tell these cells what to do.
Signals.
Yeah, the instructions. Often in older experiments, they just sort of dumped all the signals onto the cells at once.
Wow.
But natural development, it's much more gradual. It's a carefully timed sequence of signals back and forth between the cells over time, not just bam, all at once.
Okay, so it's less like flipping a light switch and more like a slow, careful conversation between the cells. So what's different about this new King's and Imperial approach?
Well, this study, it really zeroed in on creating a very specific kind of 3D material. They used something called bioorthogonally cross-linked hydrogels made from gelatin.
Ooh, okay. Bioorthogonally cross-linked hydrogels. Gelatin, that's a mouthful. Let's unpack that. Hydrogels, I get that they're like a jelly, right? A scaffold for cells.
Pretty much, yeah. Water-based, forms a 3D network.
But bioorthogonally cross-linked, what does that part mean? And why gelatin?
Good questions. So bioorthogonal basically means chemical reactions that can happen inside a living system, like with cells, without messing up the normal biology. It's like cell-friendly chemistry.
Okay.
And the cross-linking is just how they build that 3D network in the gel. Using this specific type of chemistry with gelatin gave them really precise control over the hydrogel's properties. And gelatin itself is useful because it comes from collagen, which is, a natural part of our body's tissues. Cells tend to like it.
Right, so it's building a biocompatible, very specific little house for the cells. The study also mentioned they tweaked things, like the gelatin concentration, 8% versus 12%, and the ratio of the cross-linking stuff, tetrazine and norborne. Why fiddle with those knobs?
Exactly, they were fiddling. By changing the amount of gelatin and that stoichiometric ratio of tetrazine to norborne, they tested 1:1 and 0.5:1. They could basically dial in different levels of stiffness and other mechanical properties for the hydrogel.
Stiffness, like how firm the jelly is?
Sort of, yeah. Think of it like baking. Change the ingredient ratios. You change the cake's texture. They wanted to see how that physical property, the stiffness, affected how the tooth cells developed.
Okay, so why is stiffness so critical here? Why does the firmness of this scaffold matter so much to a developing tooth cell?
Well, the stiffness and other mechanical cues from the environment around a cell, they play a huge role in telling the cell what to do, how to behave. This hydrogel acts like an artificial extracellular
The
matrix.
ECM, right, the natural scaffolding between cells.
Exactly. And it's not just passive scaffolding. It provides structure, sure, but it also sends signals. By tuning the hydrogel's properties, they could study how the material itself influences tooth development. And what's really cool here is that the specific hydrogel chemistry allows the scaffold to be, well, dynamic. It can degrade or change in situ as the cells remodel it.
Ah, so it's not just a static block. It can respond to the cells.
Precisely. Unlike older, more static materials, this dynamic interaction is probably a big reason why they got the organoids to form properly.
That makes a lot of sense. Okay, so they made these different hydrogels with varying stiffness. What did they actually do with them? What was the experiment?
They took those two key cell types, the dental, epithelial, and mesenchymal cells from mouse embryos.
Mouse embryos.
And then they embedded them together inside these different hydrogels. They also did a parallel experiment where they took whole tooth germs, like tiny, very early stage teeth, also from mouse embryos, and put those into the different gels.
Right, so two approaches, starting with separated cells and starting with these little preformed tooth buds.
Right.
What did they see with the whole tooth germs in the different gels?
It was pretty clear. In the stiffer hydrogels, the tooth germs grew more slowly, and they didn't really develop those nice, defined structures you expect to see. But, and this is key in the hydrogel with the lower stiffness, that 8% gelatin, 0.5 to 1 ratio 1. The tooth germs looked much more normal, much more typical development.
Wow, so that strongly suggests the physical squishiness of the environment really matters for the tooth germ itself.
It does. It significantly impacts development.
So what about the other experiment where they started with just the separated cells? Did they get anything tooth-like?
Yes, and this was maybe even more telling. They found that tooth organoids, these 3D structures that actually mimic tooth development.
Organoids, right, mini organs in a dish.
Kinda, yeah. These organoids formed consistently only in that same lower stiffness hydrogel, the 8% 0.5 to 1 ratio 1.
Only in that one.
Only consistently in that one. And when they looked closely with histology, they saw proper, well-organized structures with both the epithelium and the condensed mesenchyme working together. Like the recipe needs the right consistency for the ingredients to combine properly.
And in the stiffer gels, what happened there with the separated cells?
Not so good. In the stiffer ones, they mostly just saw epithelial cysts, basically little sacs of epithelial cells or just kind of undefined round blobs. Definitely not proper tooth organoids. The cells just couldn't organize correctly.
So the structure failed to form in the stiffer material. And how did they know if the cells were actually, talking to each other in that successful softer gel?
They used fluorescent markers, tagged the mesenchymal cells green. In the successful softer hydrogel, they saw those green cells nicely integrated within the organoids, mixed in with the epithelial cells.
So they were mingling properly.
Exactly. But in the stiffer, unsuccessful gels, the green mesenchymal cells tended to stay on the outside, separate from the epithelial structures. It showed a clear lack of that crucial interaction, that communication.
So it wasn't just about having the right cells. The environment had to let them connect. And the news article mentioned something about the material releasing signals slowly. Why is that important?
That's another really crucial piece. The researchers think part of the success is that this specific hydrogel system allows for a more gradual release or presentation of signals over time. It better mimics that slow step-by-step dialogue we talked about in natural development.
Right, not just blasting the cells with everything at once like maybe previous materials did.
Exactly. Those earlier materials might have just overwhelmed the cells or just didn't get the timing right. This slow, controlled aspect seems vital.
Okay, so this specific hydrogel lower stiffness allows dynamic remodeling, facilitates this slow communication seems to be the key unlock here. What does this actually mean potentially for dental care down the line?
Well, the implications are pretty exciting. This offers a genuinely promising path towards an alternative to fillings and implants. The idea of growing a replacement tooth from a patient's own cells.
That sounds incredible.
It really is. These lab-grown teeth could, theoretically, integrate perfectly, regenerate naturally if needed, maybe last a lifetime. And because they're from the patient's own cells, you avoid rejection issues.
So that science fiction idea of regrowing a tooth, This research makes it feel a bit less like fiction.
It brings it a step closer, absolutely. It's a significant move forward for regenerative dentistry and really for regenerative medicine overall. It shows the power of using these carefully designed bioengineered environments to grow replacement parts.
And it sounds like it's also teaching us a lot about how teeth form normally, too.
Definitely. By seeing how the cells behave differently in the different gels, it really highlights how critical the biomaterial environment, especially mechanical things like stiffness, is in guiding development at the cellular level. It gives us fundamental insights.
So where does the research go next? Are we talking about seeing this in the dentist's office soon, or are there still big steps needed?
Oh, there are definitely big steps still needed. The researchers are exploring different ways to translate this lab success into something that actually helps patients.
Like what?
Well, one idea is maybe transplanting young tooth cells, maybe within this hydrogel, into the gap where a tooth is missing and letting them develop in the mouth.
Interesting.
Another thought is to grow the tooth further along in the lab first, maybe to a more developed stage, and then surgically implanting it.
Both sound pretty challenging. What are the main hurdles, the limitations right now?
You're right, it's not simple. The biggest challenge is definitely that leap from making these organoids in a dish to creating a fully functional tooth that integrates properly in the complex environment of the mouth and lasts long term. Figuring out the best way to transplant it, making sure it connects to nerves and blood supply, that's huge.
Yeah, that sounds incredibly complex. And they use cells from mouse embryos in this study, right? That's obviously not going to work for humans.
That's another critical limitation, yes. They used embryonic cells here likely to really understand the fundamental development process. But for human therapy, you absolutely need a different cell source.
Like adult stem cells.
Exactly. There's other research looking into using adult cells, maybe stem cells from gum tissue, gingival epithelial cells, or mesenchymal stem cells from bone marrow. That's where future work really needs to focus for translation.
Makes sense. And the study also mentioned they didn't add extra growth factors into the mix. Is that something they might try later?
Yes, potentially. They noted that themselves. Future work could definitely explore adding specific growth factors or signaling molecules into the hydrogel itself to perhaps boost development or improve the final tooth structure.
Okay. And we heard from some of the researchers involved. Siri and Zhang from King's College really emphasized how this material gets around the limits of fillings and implants and how that slow signal release mimics the body.
Yes, and Dr. Angelova Volponi, also from King's and the lead author, she highlighted the transformative potential here for dental care, offering more sustainable, effective solutions. And she pointed out King's commitment to this kind of research.
And the paper itself really doubled down on how important the hydrogel's specific properties were, didn't it?
Yeah.
Emphasizing the role of the biomaterial in making this happen.
Absolutely. The material wasn't just a container, it was an active participant in guiding the cells. It really points towards a future where we might have much more biological integrated solutions.
So as we wrap up this deep dive, it's pretty mind-bending stuff thinking about lab-grown teeth. But let's end with this thought. If we can get this right, if we can engineer something as complex as a tooth,
Yeah?
what else could we regenerate?
Yeah.
Could similar bioengineering approaches work for other complex tissues, maybe even organs down the line? This research really does crack open the door to some incredibly exciting possibilities for medicine.
It absolutely sparks the imagination about the future of regenerative medicine. And for anyone listening who wants to really get into the nitty-gritty, the original paper is in ACS macro letters. We won't put the DOI here, but you can search for it easily. It really makes you wonder, what other questions does this raise about where dental health and even medicine as a whole is headed?
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.